Now, the next thing that we've listed are enzyme inhibitors. What's that? An enzyme inhibitor is something that attaches to an enzyme and inhibits it. It stops it from working. It stops it from catalyzing a biochemical reaction. Let's show you a picture to help you understand this. Let's look on page F5. And I wrote that. Let's see F5. So on page F5, you'll recall that we have previously described how enzymes can attach to a substrate, and the enzyme can then cause a change in the substrate and turn it into what we call products. That you needed to know for the test we've taken. But here, right below it, it shows diagrammatically an enzyme, and it shows something labeled an inhibitor. This inhibitor can attach to the enzyme, as it shows right here. And as long as this inhibitor is attached to the enzyme is prevented from attaching to the normal substrate and converting it to a product. You might say, can you say that again? Because I'm tired. All right, here's the idea. Enzymes normally attach to a substrate and convert it to something else. Sucrase attaches to sucrose and breaks sucrose apart into glucose and fructose. What an inhibitor does is it attaches to the enzyme and prevents the enzyme from catalyzing a chemical reaction. So enzyme inhibitors stop an enzyme from working. Now, many of these enzymes are absolutely essential to life. If they stop working, if they stop catalyzing a chemical reaction, you die. So if you stop certain enzymes from working, you're dead. What's an example of that? So back on the previous, where we were on F3, so I'm getting mixed up here, on F3. So on F3, what's an example of something that would stop an enzyme from working that would cause you to die? Cyanide. Cyanide is one of the most lethal substances known. It actually attaches to an enzyme. I'm not going to ask you to remember the name of the enzyme, but the name is cytochrome oxidase. I put it in brackets. Just from its name, how would you know it's an enzyme, cytochrome oxidase? The ACE ending. And this enzyme is really important. Why? This enzyme is needed for cellular respiration. Cellular respiration is the process that occurs in our mitochondria by which we make that gasoline called ATP. So what cyanide does is it stops this enzyme from working in our mitochondria. The cells are not able to make this gasoline called ATP, and every single cell in your body will die as a result. This enzyme is found in every mitochondria of every cell in your body, and if you stop that enzyme from working, you're dead. How long? 4 minutes. No antidote, no cure. You're dead in 4 minutes because you've interfered with the production of that fuel that powers all living cells. So that's cyanide. So that's why we said enzyme inhibitors can be viewed as poisons that can kill you. Another example of an inhibitor, something that inhibits an enzyme, is arsenic. Maybe you've heard of arsenic. Arsenic is a poison, a chemical that inhibits an enzyme called succinate dehydrogenase. Again, I'm not going to test you on it. I put it in brackets. We know it must be an enzyme because it ends in ACE. Interestingly, this is another enzyme also needed in cellular respiration to produce ATP. There is more than one enzyme needed to make ATP in the mitochondria. So again, if arsenic stops this other enzyme from producing ATP, you're dead. So arsenic is also quite a lethal poison. Now, there are, of course, chemicals they have discovered that interfere with all kinds of different enzymes in our body. Some interfere with enzymes in our brain cells. Some interfere with enzymes in our muscle cells. Some interfere with enzymes in our liver cells. Here's another example. Organophosphate insecticides. You'd say, yeah, like what's that? Well, obviously, if they're called insecticides, they're used to kill insects. These include malathion and parathion. Has anybody ever heard of malathion or parathion? These are commonly used insecticides. If you have little insects, aphids, on the leaves of your roses, on the leaves of your lemon tree or something, so you go to Home Depot, you go to Osh, and you say, what do you got that'll kill these little bugs that are all over my plants? My roses or whatever. So they'll hand you a can of malathion and they'll say, just spray this on the leaves of the plant or the tree and that'll kill those little bugs. Well, how does it work? It actually, this chemical interferes with an enzyme in the nervous system of those insects. The enzyme that's in the nervous system of the insects is called acetylcholinesterase. How do we know that's the name of an enzyme? It's an ACE. So these insecticides stop this enzyme from working in the nervous system of the insects, and the insects become paralyzed. They cannot move. They roll over on their backs. They say something like, "Save me!" and then they die. And that's how they work. So that's clearly, that's how these insecticides are working. They are enzyme inhibitors. They stop this enzyme from working. Now, in fact, before they ever developed malathion and parathion to kill insects, they had originally developed these acetylcholinesterase inhibitors to kill people. The most famous example of this is called Sarin Nerve Gas. Sarin Nerve Gas is 10,000 times more powerful than malathion. Spraying people with Sarin Nerve Gas is the equivalent to spraying aphids with malathion. It is, we're a lot bigger than a little aphid. So if you spray malathion on an aphid, these little bugs, that'll kill them. It probably won't kill us. We're a lot bigger. It's going to take something, either a lot of malathion or something more powerful than malathion. Well, Sarin Nerve Gas was originally developed to kill people, biological warfare. It is 10,000 times more powerful than malathion, and releasing a canister of it will basically cause people to drop dead because they become paralyzed. It works the same way. We also have that very same enzyme in our nervous system called acetylcholinesterase. You become paralyzed, you can't breathe, you can't move, you stop breathing, you're dead. And so that's what Sarin Nerve Gas is. So let me give you 3 quick examples of Sarin Nerve Gas, just because it's a bit entertaining, and so we can break up the dryness of this subject. 2 of the examples I'm going to give you are absolutely true, and then I'm going to give you one quick fictional, non-true example. I'll get to that. Okay, example number 1 really happened. So as we know, the U.S. sent forces twice into Iraq, right? There was first the Gulf War, and then later the Iraq War. And each time they expected Saddam Hussein to use Sarin Nerve Gas on the soldiers. Now, in fact, he never used it. All right? We don't know why he didn't use it, but he didn't use it. The soldiers were actually carrying an antidote for Sarin Nerve Gas with them. Now, had he ever used Sarin Nerve Gas? He had. That's not my opinion, he had. Most Americans actually do not know that Iraq actually had a war with Iran that lasted over 10 years. People whose families come from Iran do know about this. And there's strong documentation with photographs where he did use Sarin Nerve Gas in some of the towns right on the border between Iraq and Iran. They have border with each other. And there was literally tens of thousands of people that were just dead, lying on the ground, paralyzed from the use of Sarin Nerve Gas. So why he didn't use it against when the Americans went in, I don't know. Nobody knows. He did use it against Iran. That was true, and it's well documented. Don't believe me? Look it up. Google it. A second real example of the use of sarin nerve gas occurred in a Tokyo subway in Japan about 15 years ago. Some people from Japan would know very well the story, but if you don't believe me, just Google it. There was a revolutionary group in Tokyo, Japan, that released a canister of sarin nerve gas into a Tokyo subway, killing 350 to 400 people, just like they were aphids. All right? They released a canister right into the major subway of Tokyo. It's absolutely true. Don't believe me? Has anybody ever heard of it?
Yeah.
Yeah. So those are real episodes of sarin nerve gas. Now I'll tell you a fictional account. If you want to have an exciting movie to watch, especially if you like action movies, not everybody does, I do. If you want to see a good movie and you've got, Monday or no class for Veterans Day, so rent this weekend a movie called The Rock. The Rock came out in the 1990s. It stars Nicolas Cage and Sean Connery. Some of you might remember this movie. I'll tell you the synopsis of it. There's some bad soldiers, and these bad soldiers, American soldiers, decide that they want money from the government, from the United States, and so they take sarin nerve gas, which is depicted as these little green pellets. They place them on Minuteman missiles that they have positioned on Alcatraz Island. Now, where's Alcatraz Island? It's an island in the middle of San Francisco Bay. Now, the nickname for Alcatraz Island is The Rock. It used to be a place for a prison. And so that's how the movie gets the name The Rock, because that's where all the action occurs on Alcatraz Island, which is very scenic, right in the middle of San Francisco Bay. They contact the President of the United States. They demand each one wants $1 million within 24 hours. Or they threaten to launch the Minuteman missiles and release sarin nerve gas over the city of San Francisco, which is going to wipe out the entire population. So in the movie, they have somebody playing the part of the president. And they said, we're not going to go and be intimidated. So what do they do against these bad guys? They send in the Navy SEALs. Now, Navy SEALs, this was done in the 1990s, way before the more recent use of Navy SEAls, been well known for being a really tough fighting unit. So they actually come in from the ocean, from the bay. They land on the Alcatraz Island. But unfortunately, the way it's depicted in the movie, has anyone ever seen this movie? Yeah. Okay, the way it's depicted in the movie, the bad guys machine gun every single one of the Navy SEALs. Anybody remember this? So now they machine gun the bad guys, the entire Navy SEAL squadron. Now, what do you do now? You're the President of the United States. You've just lost your Navy SEALs, the toughest fighting group you've got. You got no choice. You didn't want to do it. You have no choice now. You've got to send in Nicolas Cage. So Nicolas Cage, who plays the part of somebody who's a toxicologist, an expert in poisons for the FBI, and they're dealing with sarin nerve gas, which is definitely a poison. So he goes in along with Sean Connery, who's like about 107 years old, and nobody knows what he's saying. And it's too complicated for me to even begin to explain what he's doing in this movie. He plays the part of a British secret agent who was the only person to ever escape from Alcatraz Island and knows how to get on there without being detected. But they go in, and the good news is they end up killing all the bad guys. Near the end of the movie, Nicolas Cage is wrestling with the one last bad guy left. He's got this sarin nerve gas pellet. The bad guy kind of shoves it in the face of Nicolas Cage. Nicolas Cage turns it around and shoves it into his mouth. He collapses, paralyzed, but Nicolas Cage has been exposed. If you remember the scene in the movie, he's actually carrying a syringe with the antidote, and there is an antidote. He pulls the syringe out, and he injects it into his own chest. The way the antidote is given, and this is real, is it is injected directly into the heart, because you've got to get it through the bloodstream as rapidly as possible, or you're dead. The only problem is, in real life, he would have already been too paralyzed to give himself his own injection into his own chest. But we didn't want Nicolas Cage to die. It's a good action movie, and it shows you, the whole thing revolves around sarin nerve gas. All right, anyhow. So far, what we've learned about chemicals that inhibit enzymes is they can kill things, whether it's people or whether it's insects. So we might ask, is there any benefit to this stuff? I mean, fine, we're learning very nice, clever ways to kill things, kill people. So like everything else that we've ever learned, everything that we learn in science and technology can be used for good or bad purposes. So the scientists who've developed sarin nerve gas and other enzyme inhibitors are thinking, they're thinking. Let's see. Okay, so we got chemicals that can stop enzymes from working. What if we found a chemical that could stop an enzyme in bacteria from working? If you can stop an enzyme that's found only in bacteria, but not human cells, from working, the bacteria would die, but it would have no effect on the human cells. Does everybody follow what I just said? That's how all antibiotics work. The very first antibiotics ever developed were developed in the 1930s. The very first ones are called sulfa drugs. They're still used. There's better antibiotics than these in general now. One of them that's still used goes under a brand name Bactrim and Septrim. What it does is it interferes with an enzyme found only in some bacteria. It's not found in human cells. You certainly do not need to know the name of the enzyme. It's called dihydropteroate synthetase. And we know it's an enzyme because it ends in ase. And literally, when you swallow an antibiotic, you are swallowing an enzyme inhibitor. Something that inhibits the enzyme in a bacteria that's in your body. Since what you're giving is something that inhibits only the enzyme in the bacteria, but not in any enzyme in a human cell, it has no effect on the human cells. It only kills the bacteria. Now, the sulfa antibiotics were developed in the 1930s. Shortly after, in the beginning of the 1940s, is when they discovered penicillin. And after penicillin came erythromycin and all the others. Think about this, just to reflect on history. Before, we've touched upon this before, imagine, before the 1940s, before 1930s, there was no antibiotics. When somebody got sick, you know what usually happened? They died. That's what happened. All those childhood infections, how many times have you had an earache and you went to the doctor, they gave you a prescription for amoxicillin or erythromycin, and a few days later you were fine, the fever was gone, the pain from the earache was gone, and you forgot you were ever sick. Just now pretend there was nothing to give you. So that made all the difference in the world. So understanding inhibiting an enzyme not only was, unfortunately, how they created sarin nerve gas, but it was also the beginning of how they learned to kill bacteria and use what are called antibiotics. And if we think about what the word antibiotic means, it literally means against life, against a living thing. It stops a living thing from working by stopping that enzyme. Now, at the bottom, we remind you that enzymes are proteins, arguably the most important of all proteins, and we have learned back in section D that there are a number of things that can cause proteins, whether they're enzymes or any other proteins, they cause them to unravel, to denature, to uncoil, and stop working. So we know that high temperatures, hyperthermia, having a high fever can lead to death as the proteins, including the enzyme proteins, unravel and stop working. We know that acidosis or alkalosis, an extreme change in pH, acidity or alkalinity, can cause proteins, including enzymes, to unravel and stop working, resulting in death. And we know that certain heavy metals like lead poisoning and mercury poisoning cause these proteins to unravel, including enzymes. And that takes us to page F4. On F4, we have learned so far about two major types of biochemical reactions that are associated with energy. One of them is photosynthesis. And as we remind you, we learned this in section E on the cells, when we're learning about chloroplasts, that photosynthesis occurs within the green chloroplasts in plant cells, and it's called photo because it uses light energy in order to synthesize, photosynthesis, to synthesize sugars and other organic molecules. Now, for those of you taking lab, the next couple of terms you probably have already learned in lab. If you're not taking lab this semester, I assume you'll be taking it next semester. I'll remind you, until you take the lab part of the class, you cannot transfer a lab science because you've taken maybe the lecture part but not the lab part. Organisms that carry on photosynthesis, i.e., that is, they make their own food, are called autotrophs. Auto means self and troph means to feed. So plants are obviously autotrophs because they make their own food using light energy to make sugars and other foods. Organisms that do not carry on photosynthesis, i.e. that is, they do not make their own food but must consume, ingest, eat other organisms are called heterotrophs. Now hetero means other, troph means to feed. So to feed on others. And now you've seen that Greek root hetero is heterosexual. A heterosexual is somebody who is sexually attracted to the other sex. So hetero means other, heterotroph means to feed on others. So how would we describe us? Are we as humans autotrophs or heterotrophs? Heterotrophs. Or heterotrophs. We have to eat other things. We can't make our own foods standing out in the sun. So we eat other things, and everything that we eat, we may have mentioned this before, with the exception of water and salt, came from a living thing. Whether you're eating meat, came from a living thing, an animal, eggs, milk. Whether you ate salad, lettuce, tomatoes, cucumbers, squash, you're eating living things, things that were alive. And that's how we get our nutrients. Now, photosynthesis is one process that involves using energy, light energy, for plants or autotrophs to make their own food. The other process that we mentioned in section E is cellular respiration. Cellular respiration is the process by which organic compounds, usually sugars like glucose, but other organic compounds, including even fats and even proteins, can be broken down, releasing energy, and that energy that's released is used to produce that gasoline that powers all living cells called ATP, this high-energy nucleotide. Now, what I wrote next is obviously important. It's important because it's all in bold print, it's underlined, and it's in a larger font. So I guess I want you to know it. So if you don't know this for the test, I've done everything I could to convey to you its importance. Cellular respiration occurs in all living things. It doesn't matter whether we're talking about plants or animals. It occurs in autotrophs and heterotrophs. One of the major distinctions between plants and animals, and obviously there are many, is how they obtain their food. Plants make their own food. They are autotrophs. Animals, including us, have to eat other things. We cannot make our own food. We are heterotrophs. We feed on other things. But once an organism has food, either having made it itself, as in the case of plants, or ingested, consumed another organism, all living things break down the food, releasing energy in order to make ATP. So cellular respiration is a fundamental process that occurs in all living things, plants as well as animals. So I hope you got that. Now, what I've listed next are two of the 4 laws of thermodynamics. And we'll just go through these real briefly. These are scientific laws that nobody disputes. We know the closest that science ever gets to feeling that they have described something that's fundamental, they elevate to the status of a scientific law. The first law of thermodynamics says that the total amount of energy in the universe is constant, although energy can be converted from one form into another form. So, for example, if you have a flashlight, what powers your flashlight is a battery, and in that battery is stored chemical energy. When you turn on that flashlight, that chemical energy starts to be converted into electrical energy, because there's actually an electrical current moving through a wire that goes to that little light bulb in that flashlight. And then, as that electrical energy goes through the wire, the tungsten filament wire in the light bulb, it causes the electrical energy to be converted a third time into light energy. So literally, when you turn on a flashlight, chemical energy in the battery is turned into electrical energy that flows through the light bulb, and the light bulb converts the electrical energy into light energy. So clearly, one form of energy can be converted into another form of energy. That's basically what the first law of thermodynamics says. It predicts you can do that. The second law of thermodynamics, however, says the following. Whenever you do try to convert from one form of energy into another form, some of the energy goes off as heat. This is known as the second law of thermodynamics, or the law of entropy. The law of entropy. So what's an example of what I'm saying? Let's look at the light bulbs right above. So the light bulbs right now are converting electrical energy into light energy. But we know that light bulbs get hot. So some of that electrical energy is being converted into heat, which is a form of energy, heat energy. That's not why we have light bulbs. The reason why we have light bulbs is not to warm up the room. But we know that when we convert one form of energy to another form, electrical into light, some of that electrical energy is going to be converted into heat. And there's nothing that we can do about it. It is impossible to have 100% conversion of one form of energy into another form. Some of the energy is always going to end up as heat energy. The way this has been somewhat cleverly stated is the following. The first law of thermodynamics basically says you can't get something for nothing. If you want light energy, give me electrical energy. And then we can convert electrical into light. But the second law of thermodynamics is somewhat cutely stated as you can't even break even. In other words, you can't get something for nothing. And in fact, you can't even break even. Because when you do convert the electrical energy into light energy, some percentage of that electrical energy was turned into heat energy, even though that's not what your intention was. So we're going to come back to these ideas, but you should be familiar with the first and second laws of thermodynamics.